All right, on to the primitive streak. So we just got done talking about gastrulation. Now we'll talk about what kind of marks some of these events of gastrulation. Here's my Da Vinci gastrulation image. Wonderful. Okay. So primitive streak. Let me get my pointer out again. So we are here in gastrulation again. And so the primitive streak is kind of like the gateway or the start of gastrulation. So it's a crucial structure that forms during the early embryonic development in amniotes. So this is, remember, we kind of talked about the amniotic egg, I believe, earlier in a lecture. And so reptiles, birds, mammals, anything, vertebrates, not all vertebrates, because fish wouldn't be, or amphibians aren't included in this, but reptiles, birds, and mammals. So it represents one of the first visible signs of gastrulation, because we can actually see the process without staining or anything like that, just with like a microscope. So this process transforms a simple ball or disc into a cell of cells into a complex multi-layered embryo. So that's, you can see this is kind of forming right here. And then it forms along the midline of the epiblast, so kind of, straight down the middle. The upper layer, this is the upper layer of the embryonic disc, and it appears as this like thickening long line of cells. We'll get into some images in a second here, and this happens about 14 days post fertilization in humans, but it happens sooner in animals that don't take as long to develop, like humans taking 9 or 10 months. Chicks and things like that take much shorter, so that 15 days is pushed forward. So this is, we're going to look at it from the point of view of a chick egg. Like I said, we know we can't study as easily human embryos, so a lot of this work is done in other things. So we'll talk about the chick egg. So here's a good reason why we looked at those different ventral, dorsal, and stuff, so you can kind of orient how these pictures are going. So ventral being from the front, dorsal being from the back, and mid sagittal is from the side view. So this is during chick development. We have this Kohler's sickle is a area where we start to get this local thickening of the epiblast tissue. So we can see the areopeca of the epiblast here. The area opaca is a peripheral ring of the blastoderm cells that have not kind of shed their deeper cells yet. So if we were to look, let's go back. Where's my pointer? So they haven't shed their deeper cells down here, right? So we have, it's kind of this layer going right there. The area pellucida is part of the blastoderm where the inner cells have kind of detached, right? So we have opaca, so probably, I'm assuming, comes from being opaque, so darker because it's got like a layer underneath it. Area pellucida, there's not this second layer underneath it, right? So it's a one cell thick layer, I guess is the best way to put it. A single sheet as opposed to multiple layers down. Okay, so at stage X here, we have these little hypoblast islands, and they also have these precursor cells that are around the Kohler sickle here. By stage 12, so a couple stages later in development, a sheet of cells grows anteriorly from the sickle, and these hypoblast cells start to connect here, and this migrates across the ventral side of the cell, or ventral, I shouldn't say cell, of the embryo, the ventral side of the blastocle. And then by stage 13, just prior to this formation of the primitive streak, the formation of this hypoblast has been completed. So we get like the second layer all the way across here, right? And this is just on the ventral side. You can see it's not on the dorsal side. So now when we move to stage 2, this is about 12 to 14 hours after the egg has been laid. So it's been in the chicken for a while, but this is after it's been laid. The primitive streak cells form a third layer that lies between the hypoblast and the epiblast cells. So we are starting to form this middle layer. And then by stage 3, which is about 15 to 17 hours past laying of the egg, the primitive streak has become a definitive region. So this layer starts to move across the ventral side here of, or the ventral side of the blasticle, or the gastrula, I guess would be a better proper term to call it at this point, since we're undergoing gastrulation, the gastrula, and you can start to see its formation. So this is kind of just a view of how this happens at a little cellular level, but we're going to zoom out to kind of see what this looks like here in these next slides. So anterior, posterior, right? So head and tail. Oops, I wasn't circling with my right thing here. We can see the area opaca is this kind of darker layer here. The area pellucida is the inner, and then this colar sickle, that's kind of where all this starts to emerge from, and that was that kind of densely packed area of cells, right? So cell movements of the primitive streak is what we're going to look at here. So this is the blastoderm if we look at 12 to 14 hours. So you can see where we can notice here where colar sickle is and where the streak will emerge. So this is kind of our starting point right there. This is 15 to 17 hours, and this is still the dorsal view and starting the elongation. So that's those cells that are replicating from the hypoblast. They're near the primitive streak. It's starting to take shape. So it starts at colar sickle. It doesn't move like generate. It kind of like stretches out or moves from that location, right? So this is 18 to 20 hours after the egg is laid, and we can start to see that we get at the top of this primitive streak, we get what's called Henson's node. We still see this area pellucida and the area opaca surrounding it, but this primitive streak, we now, since it's opened up, we would call that the primitive groove. And this is just a depression or opening that forms within the streak. And this is where those migrating cells, so we saw those different migration strategies earlier, this is where they'll migrate through. This provides access to the inside of the developing embryo. So cell movements of the primitive streak, I'm reading the title again. Formation of the notochord and the mesoderm somites are shown here. So we have Henson's node again, and we have the head process up at the top. The center of Henson's node, or the center of Henson's node up here, forms a pit where cells can enter the embryo and form the notochord in the prechordal plate. So we'll get into the neurulation stuff a little bit further going on, but that all happens. So this is an important feature to keep in mind, Henson's node up there. And then, I guess let me, as the primitive streak proceeds, it will move across this ball of cells, right, to put it bluntly, and then it will retract. It'll, I guess retract is the best word, or regress, or come back, right? And as it does, it pulls and lays down this the notochord in its place, right? So as you can see, we have here the notochord being pulled from the opposite end of where the primitive streak started. So it'll like open up, allow those cells to migrate in, and then pull back to form this notochord. So this is 23 to 25 hours after the egg has been laid. And then this slide here, oh, I didn't write down the time here. But so the notochord's been laid down here, right? So we can see that it's kind of faint, but it's a line that is replacing, or it's kind of zipping up, I guess is the best way to say, like the primitive streak there, right? And then on the different sides at the top, we have the neural fold. In the head fold, this will develop into the head. And then we have the notochord, which would be the spinal, or well, where the neural tube will be directed to develop from. The notochord directs the development of the neural tube, which is kind of above it, and then to the sides of it are the somites. So remember, that's the ribs and stuff. So you can kind of see how this is shaping up, where we have the head up here, the primitive streak starts down at your kind of at your tailbone, it goes up, stops at the where your head's going to be, then lays down, zips back up, lays down the notochord with it, or kind of pulls the notochord along with it, which directs where your neural tube and your spinal cord is going to be. The somites develop off of this notochord. That's where your ribs and things are going to be of that nature, kind of your midsection here. So if you squint at it, you can kind of see the organism that's being developed here. Okay. So this is the 4-somite stage, so we can actually count the number of somites there are on each. So you just count one side. There's 8 here, but there's 1, 2, 3, 4 somites here. So that would be a stage that a developmental stage. A lot of counting of cell numbers or somites or developmental milestones, the gastrulation or the primitive streak being present, the notochord, etc. So this just kind of shows the migration. This is like the cell disc, where we have the migration of these cells in and out through the primitive streak here. So we have Henson's node up at the top. So we have the blasticle, which is the kind of inside area, right? The epiblast is the cells of the outside here. And we're starting to form our cell layers. We have these cells migrating in from the exterior, from the epiblast, to be mesoderm cells. The endoderm cells are already in place here. They're on the inside of the blasticle, so that's the pocket here. And we have the hypoblast cells that helps to start this whole formation off. Let's see, let's look at my notes, make sure I didn't miss anything. The hypoblast cells eventually sort out to form a layer beneath the endoderm and contribute to the yolk sac. So they're actually not really used for, they don't turn into structure like the other 3 layers do. They contribute to the yolk sac. So cells migrating through the node travel anteriorly to form the precordial plate and the notochord. So that's the ones that migrate through Henson's node at the top will go up, so the orange here, to become the starter cells for the notochord. So remember, the primitive streak would start down this way, come all the way up, kind of interact with those cells that migrated through Henson's node and are becoming the notochord, and then zip it back up as it pulls it down posteriorly. So the ones, let's see. These purple ones that migrate through up here become the somites, and the red, the cells that are navigating this region further posteriorly, become mesoderm tissue. Okay, there's a lot of information in this slide, so I'm making sure I cover everything. Mesoderm. Yep. Well, so for the mesoderm, we have mesoderm and we have intermediate mesoderm. So mesoderm, there are different kind of layers to the mesoderm, or or gradient to the mesoderm, right? So remember with like fate mapping, depending where you migrated through and where you're positioned, you'll develop into different things. So if you are further anterior, you'll develop into higher up tissues, and if you think of it from an adult body, and if you are more posterior, it'll be lower tissues, right? So while we kind of just all group them into mesoderm together, where they migrate through still dictates what kind of tissue they're going to become, or is correlated with what kind of tissue they're going to become. Okay, so this is just kind of we're looking at a actual real one, micrograph, I guess is the best way to put it, instead of a figure or an image or a drawing, I guess. So you can see the area opaca here, the area pellucida, so opaque, darker, pellucida, easier to see. You can see this primitive streak formed up here. We have the head process, which is kind of Henson's node. So you can see it's gone up and it's starting to come back down, and you can see it's pulling this part along down it, and that's the notochord. So this is the 2 somite stage, which is about 25 hours for the chicken embryo. You can see the where is it? Oh, here we go. The pharyngeal endoderm. The primitive streak is regressing, so it's moving back. Regressing is a good word. When I say retreating, so getting pulled backward or going backwards, leaving the dorsal axis of the embryo, including the notochord. So here again is Henson's node. It's getting pulled back. We're having the head process and the notochord laid in its place. So it's unzipped. It is a lot like a zipper. So like you have it unzips all the way up, so it opens, allows the cells to come in, and then the zipper gets pulled back down, and it pulls the notochord down with it. So it's laid in a head to tail direction, and it starts at what would be your ears, right? Because that's kind of where your spinal cord attaches, the hind part of your brain, the primitive part of your brain, and it goes to your tailbone. So here we have 24 to 28 hours, or the 4 somite stage. You can see them here in this picture really good. So these are those little pockets of cells right next to the notochord that's been laid down. The primitive streak is almost all the way down back to the tailbone, and we have this the notochord here. We're starting to form the neural groove, so that will eventually become the neural tube. So it starts as a groove, like a divot, and then it turns into a tube all the way along. We're getting the development of the foregut, so that would be the upper gut, right? Your stomach and esophagus, etc. And then here we are 28 hours, and so it is regressed all the way. We're at I would call this 1, 2, 3, 4, 5 somite stage, and it's closed all the way up. So the primitive streak is gone. We have a notochord all the way, and we've kind of created our mirror plane, right? So we have mirrored on the left and right at this point is section, right? So we're going to be, besides things like livers and things like that, there's stuff that isn't mirrored in your body, but a lot of it is mirrored, right? So we've created that plane, your backbone plane. So this is just kind of a graph of what's going on here. So you can see we have the notochord, or Henson's node up here, and oops, I gotta use the right clicker here. Henson's node up here at D, and then as it regresses over time, so you can see this is kind of the time on this end, that we have the lengthening of the notochord and the shortening of the primitive streak and how they're kind of tied together, right? So as this gets pulled down, the notochord gets longer over time, and at about 20 hours is kind of when this process stops. 28 for the chick. Wrapping up, this is the last slide, but I want to make sure I've kind of covered everything. So the significance of this, so the primitive streak, again, serves as this migration pathway for cells from the outside to form the inside layers. In this also is the mechanism or start of how we get our 3 layers, the ectoderm, the mesoderm, and the endoderm. So those 3 different tissue types are what become the 3 different, we have like external layers and internal layers, right? And then it establishes the axes, both. I should have mentioned this earlier. Not only does it establish your left and right axis, right, your mirror plane, but it also is what establishes your head and your tail, right? Because Henson's node goes up to the top there, and that's what forms, it starts the notochord, pulls it down, and that's what, let me go back to, we get the head folds being started, and the neural folds, and the foregut, etc. So it's, that process starts not only the left and right axis, but your heads and tails, anterior, posterior axis as well. Symmetry breaking, okay, so yep, it helps form the symmetry, like I said. I'm pretty good at just getting this off my head and then, or out of the top of my head, and then seeing my notes and being like, nailed it. And yeah, that's about it. I think I covered everything. So that's primitive streak. Primitive streak begins, or starts this left-right axis, posterior-anterior axis, and gastrulation, and the laying of the notochord, which will eventually lead to the development of the neural tube, etc. So that's kind of the 1,000-yard view. So thanks for watching. What did I keep this at? 21 minutes, so not bad for a process this complex. All right, thanks for watching. I hope you learned something, and I'll catch you in the next video.